Hi, welcome to Detours, Understanding Acquired Brain Injury. Unit 2, Chapter 7. Oh, baby. All this seems so straightforward, doesn't it? Of course not. It's biology. What do you expect? Let's look at how eggs and sperm develop beyond meiosis, but we're talking about how they become more than just a chunk of haploid DNA, but how the actual eggs, sperm develop and what happens with the fertilization process. Now, it's true the process of meiosis helps to provide the genetic diversity, but each sperm and each egg is unique genetically. And so it's a throw of the dice. It's hundreds of millions of sperm and tens of thousands of eggs that could meet up. And so it's a huge amount of probability, not to mention epigenetic factors. Each time yields a very unique result. All right, so sperm cells develop in a process called spermatogenesis, which is mostly what we discussed. Sperm are pretty simple. You get 4 cells from one progenitor. You get 4 sperm out. With eggs, you get 1 viable egg out of one progenitor that becomes 4 haploid cells, but only 1 becomes a fertilizable egg. Let's go through, okay? So first, the sperm takes 70 days to develop and progress from a single diploid cell to 4 mature haploid cells, okay? The sperm grows a tail, which is referred to as a flagella in biology, and this is powered by the mitochondria of the sperm cells. This development, these changes are part of the genes that are in the DNA, and these are conserved, because if they're not, that sperm will never get anywhere. So somewhere in a man's DNA, in the Y, is instructions for producing sperm tails. And this is what happens to the male mitochondria, for why they don't pass them on, because those mitochondria are used to drive the flagella. This is why your mitochondrial DNA comes from your mother. All right, so there are some thoughts that maybe fragments or pieces of male mitochondria might make it, but this is all speculative, so we're not going to head down that path. Anyway, a sperm is small and primarily a haploid nucleus. That's it. It's able to, so basically it's a swimming haploid nucleus. Mother Nature's parsimonious. She'll put the least amount of effort that she can to get what she needs done. That's always been true when it comes to biology. You see that again and again. She'd rather try a million times to get something rather than, try to make it just right. That's how biology goes. All right. The egg, on the other hand, it follows a more circuitous route. Sperm-like guys are simple. Yep, yes we are. So let's talk about what happens with oogenesis, or development of egg cells. This is kind of interesting. The germ cells, the egg, divide initially, but they only go through part of the meiosis process. They're to build up their total numbers and developing in the ovary, but they are not ready to go through the complete meiosis process. Developmental arrest occurs with the egg, prophase 1. Egg cells, primary oocytes, remain in stasis, kind of asleep as it were, till the process moves forward after the young lady enters puberty. But, while she's still developing, even while she's in utero, the girl, her germ cells, she produces the total number of eggs she's going to have, but it doesn't go any further than the initial mitotic division. All right, so secondary oocyte and first polar body. All right, after puberty occurs. This applies to any placental mammal. It completes meiosis 1 based on hormonal signaling. So the first movement to the next stage occurs when estrogen and progesterone pass through the body. Okay, this results in an unequal division, which causes a large secondary oocyte, which is the structure that will support the egg. That this large cell isn't actually the egg. It is meant to support it. And then there's a small structure called a polar body, which is disposed of, but it is haploid. And those start to develop along with the oocyte, the large structure that is going to support the egg. Massive, many times bigger than the sperm. But that structure is not the egg. The egg's inside. All right, so then as the eggs start to develop, start to move through meiosis, the first polar bodies will disintegrate. And there will be other oocytes that develop and first polar bodies disappear. Now, arrest at meiosis 2. As you're getting ready to go into meiosis 2. The secondary oocyte arrests again, meiosis 2 during metaphase. So as they're getting ready to... This process of meiosis 2 will only continue if the secondary oocyte is fertilized by a mature sperm cell. So it breaks apart, and so we've gotten these structures that aren't really fully developed yet. We've got the oocyte, the really large structure that has, but it's still not complete yet. Okay, so the follicle will rupture and release this structure, but that's not quite finished yet. Okay, so what happens then? After the follicle ruptures, releases the oocyte. If a sperm penetrates the oocyte, this structure holding another cell that's kind of in arrest, this will become the egg that is fertilized. If the sperm penetrates the secondary oocyte, meiosis 2 will then complete. So once the sperm, because it's a big distance for microscopic sperm, once they penetrate the oocyte, then it will trigger the egg, which is inside the oocyte, to complete development. And that is the ovum, or the egg, that is inside the oocyte, that big structure. Now, first of all, the sperm will send a signal, and it will change the structure of the oocyte so no other sperm can enter. And the sperm will eject its tail, and the egg, inside the oocyte, will meet up, and it will combine the haploid instructions of the egg, the haploid genome of the egg, and the haploid genome of the sperm together. Now you've got a fertilized diploid zygote. That is what it is called, a new individual. You've got the cells combined, mom, dad, each parent. Now you're ready. And then the zygote will very slowly begin to divide. It takes between 12 and 24 hours for each cell division. And eventually, this destroys the oocyte, which provides support, nutrition support, and all that kind of stuff. And then there is a second polar body, which is haploid, which is also disposed of. 2 polar bodies, 1 oocyte, 1 egg. 4. 3 are lost, 1 haploid structure, just fertilized, 1 egg, 1 sperm, new individual. Now the thing is, sperm cells only have 24 hours in which they can fertilize a cell. So, 4-stage process specifically. Sperm cells become active and more able to interact with the egg, hormonal signaling. Sperm and egg recognition and binding. The sperm must be able to recognize the egg and bind to it, usually in what's called the zona pellucida, which is a layer of the egg that protects it on the outside. It's important to understand that, it's like this will allow or disallow. You've seen these videos. Those videos where sperm trying to get into the oocyte, where it needs to basically get through that outer layer, because then it can turn a chemical switch which prevents other sperm from getting inside. Because if they do, then you will have not just diploid, but triploid or quadruploid cells, which would be non-functional. So it's really important that at least 1 sperm gets in there and no more. All right, so with the fertilization of the secondary oocyte, the egg, the process of meiosis 2, it's over, and now we can start mitosis. The zygote forms, and we start to divide. Finally, with the zygote, it's unique. Its genome is unique. It is a new individual, and we'll begin to divide. As I said, each division takes between 12 and 24 hours. And initially, because the oocyte's so big, it'll be a couple divisions over days and days and days before it ruptures out of the oocyte. And meanwhile, the zygote is inside this. It travels down from the fallopian tube down into the uterus, where it will implant into the uterine wall some of the part of the structure of the zygote will begin to form into the placenta, which will support the function of the embryo. It's not called an embryo yet. That will happen about a week or so when certain structures form. That's embryology. That is a whole other field. We're not getting into that right now. But that structure forms too. Once we're implanted, then growth follows based on the genes inside the embryo. It follows its own path. And they're the same genes that are also inside the placenta, which also has genetic instructions that it follows. And so, like I said, that's its own thing. All right, if you made it this far, you should pat yourself on the back. This stuff can be quite complicated, but it serves as a solid foundation for understanding. Next unit 3, we will be getting more into some of the abstracted laws of genetics, Mendel, and how about dominant and recessive traits, things like that. But you need at least some kind of understanding of the mechanics of how proteins are produced and how cells divide and things like that in order to make sense of these kind of things. So when you hear this terminology, you're like, I know what that is. That's what this was all about. So, we'll see you soon. Please click like and subscribe. Don't forget the bell icon. Leave any comments or questions you may have. Please share with anyone who you think may benefit from watching. See you next time. Unit 3, Chapter 1. Bye.